Okay. Morning, everybody. How's it going? Let's see, I got that. Yes. Yay, people are awake. Yay. Morning, everybody. Aw. Makes me happy to see people saying good morning. It just makes me know I'm not just like a weird person, like alone in a room with my computer talking to my computer. Because it feels like that sometimes. All right, let's share this. Cool. Gosh, I can't believe that we're in the conifers already. It's a little crazy because there's only, so it's like conifers and then there's angiosperms and then that's the whole class, the whole course. So it's just amazing to me to be this far into the course. Where did the time go? It went away, I guess. But yeah, we've really made our way through the plant kingdom, make this big, which is pretty incredible, which is a pretty incredible thing. And also good on you guys. You've hung in there listening to me talk about sporophytes and gametophytes and all these other things, phylums. So good on you as well. All right, I think I'm gonna go ahead and get started. Let me just double check this. Yes. Okay, so there was an assignment due today. Hopefully you guys did it. I know you were probably annoyed that you had an assignment due today, but part of the reason it was due today is because I need time to grade it, because exam 2 is 2 weeks from today. Oh my goodness, 2 weeks from today. That's something in the chat box. Yes. Which is incredible too. Wow, 2 weeks from today, we have a lecture exam 2. So that's why I got to keep moving us forward with these assignments so that you guys have an opportunity to see the material before before the exams, but also just to keep you guys with it, right? Like you need you guys to stay with the material and don't get too relaxed. And you had spring break, so you had a little bit of a break. Okay. Yeah, so lecture exam 2 is 2 weeks from today. There will be a study guide available on Blackboard on April 1st. And no, that is not an April Fool's. It will for real be available that day. So check it out. And the lecture exam 2 covers everything from the ferns to the conifers, which is today's class. So hopefully we get through the conifers today, and then that will give you guys 2 whole weeks with all the material to study and whatnot. Yeah, if you did not turn in lecture, come on, tongue, get with the program. If you did not complete assignment 5 before class today, please send me an email so I could reopen the assignment for you so you can get points. All right. You may notice a different venue here. I'm in a different part of my apartment this week because my parents are coming to visit, and so I've transformed my office into a bedroom for them. So hence the new location. No, I'm not like on the lam or something, or I'm not like in some faraway place having vacation. Nope, I'm just in a different part of my apartment. All right. So last time we covered ginkgo, and today we're going to cover the last of the gymnosperms, which is the conifers, or the phylum Coniferophyta. And man, this is what I was saying before. Look, whoa, so we're in the vascular plants. The non-vascular plants we've left far away. Spore-bearing plants, all the way in the past. We've been dealing with the gymnosperms in the last few classes. We've already talked about cycads. Last time we talked about ginkgo. Today we're going to talk about the conifers, and then the remaining classes are basically dealing with the angiosperms, and that's it. We've gone through the whole plant kingdom. So conifers. Okay, so they're in the phylum Coniferophyta, which is nice. it's always nice when the phylum name is like the common name, right? Like when you were in the liverworts, it was Marcantiophyta, right? And your hornworts, it was Anthocerophyta. That's a mouthful. And yeah, Coniferophyta is a mouthful too, but at least it has conifers in the name, right? We saw that too. Actually, the last several classes have been like that, right? We had the Cycadophyta, you know, that has cycads in it. We had Ginkgophyta, that has ginkgo in it, and now Coniferophyta, conifers. So that's always nice. All right, conifer. Conifer comes from the Latin conifer, which means to bear cones. So bear or have, right? To have cones or bear cones, make cones, carry cones. And that comes from fere, which means to bear, and conus, which means a cone. So only conifers have cones. Only conifers have cones. I know sometimes the book, if any of you have even pulled up the book anytime this semester, maybe even the lab material, I'm not sure, might say that other things have cones. No, only conifers have cones. It's in the name, conifer, to bear cones. So that's who has cones, conifers. So if I ask you a question and say, oh, which of these is true? And one of them is like, well, cycads bear bigger cones than conifers. No, because only conifers bear cones, right? So here's some examples of cones in this picture on the bottom. Usually kind of a similar look in general, right? Kind of a woody thing. When they're matured, they're brown. But they can be all different sizes and to some extent all different shapes, but not as diverse as the strobili that we saw in the cycads, right? And these are not strobili, these are cones. Strobili, I think, do I go into it? No, maybe I don't go into it. Let's see, do I? Let's see. Yeah, I think we'll get there in a second. Yeah. But anyways, these are cones, these are not strobili, only conifers have cones. Well, we're in the vascular plants, so we know that the sporophyte generation has to be dominant and long-lived. We know the sporophyte generation is vascular and has true roots, stems, and leaves. We already know this. Conifers are big, usually big, large trees, and they always pretty much have abundant wood and bark. And we'll talk about bark later today because we haven't talked about bark yet. So in the world right now, of all the living plants that are in the world right now, the largest trees in the world are conifers. The largest trees in the world are conifers. Here is a little schematic to show you some of those large trees, So for example, let me annotate because I love annotating. Oh, do we have a question? Okay. Please make sure you guys mute. Unless you have a question, happy to answer any questions. All right. So do you see the schematic here? All these trees on it? I want to draw your attention to the far right corner. So do you see this little dot in the far right corner? That's supposed to be a person. So that little dot on the far right corner is supposed to be an average size person, right? Let's say like 5 to 6 feet tall adult, right? 5 to 6 feet, maybe a little bit, average. We'll tie average here. So with that in mind, look at these trees. Right? So let's see, some of these occur in our region. For example, Douglas fir, which we'll talk about next time, can get up to 250 feet tall. Look at that compared to this little person on the right? Wow. But the largest trees that are still alive and occur today are these, the sequoias and the redwoods, right? Which can reach almost 400 feet tall, but between 300 and 400 feet tall. So it's really, really big. It's really, really, really big. but you don't have to take my word for it. Like, here you go. This is the tallest living tree in the world right now. It's in California. Its name is Hyperion, and it's roughly 400 feet tall. It's an example of one of these coastal redwoods or Sequoia sempervirens. And again, look, here is a person sitting next to this trunk of Hyperion. Look at how wide this trunk is. And honestly, this isn't that wide considering how tall this tree is, but it's still pretty wide. Gives you an idea of the type of growth that occurs, right, in these trees, right, when they're wide. Well, bells should be going off in your brain. And if not, go back to the other lectures. But here you go again. Wow. So you don't believe me, right? You're like, yeah, okay, it's big deal. It is big deal. It's big, super big. Look, here you got Big Ben. This clock tower on the top, on the right, is called Big Ben because it's big, right? But not as big as Hyperion. There you go for you, for us New Jersey, New Yorkers. There's the Statue of Liberty, and hello, Hyperion is bigger than that too. So very big tree. Bet you didn't even know. There were trees that big. So conifers are not only the tallest trees in the world, conifers are also the oldest trees in the world. So there's some bristlecone pines, or Pinus aristata, that are also in California. California's got a lot going on for it in terms of plants. And some of these plants are more than 4,500 years old. So, yes. Now, not all of the, on these old trees, not every part of the tree is still alive. But of the parts of the trees that are still alive, well over 4,000 years old, which is pretty incredible. And so scientists are using trees like this to kind of understand aging or senescence in other organisms, right? We want to know, this thing has lived for 4,500 years. How? What's going on metabolically? How is it using resources? What allows it to get to that grand age? And so scientists are using this tree these trees, bristlecone pines, to understand senescence or aging. All right, so the conifers are decently big, not super big in terms of species number. There's about 600 species worldwide. Most of them are evergreen. Evergreen means the leaves stay on in the wintertime, or so they're evergreen, or the leaves stay on all year long. And each leaf can live for several years. So in terms of evergreen, for your phenology assignment, some of you, I don't know, I haven't seen everybody's photos yet, but maybe some of you have chosen an evergreen tree to monitor during your phenology assignment, right? Meaning it started green with leaves on it at the very beginning of your observation period. Well, if that's the case, probably not too much is going to happen in the month that you're observing it, but because it's evergreen and the leaves stay on year round. So there are certain areas on Earth that are dominated by conifers, meaning there's certain areas on Earth where the main plant that occurs is conifers. And the area on Earth that occurs is called the Circumboreal Forest. Circumboreal Forest, which occurs in the northern latitudes. So here is a map of the Earth. Looking down on the Earth with, here I'll change colors, with the center is the North Pole. So you're looking down on the North Pole. And this shows you where those circumboreal forests occur, right? Oh, so Ben says, I learned in my ecology class, it used to be called the Arboreal Forest of Christmas trees. Okay. I never heard that, but perhaps would make sense, which Ben likes better. The Arboreal Forest of Christmas trees. All right. Well, yes, fine, Ben. But you should still remember Circumboreal Forest. And I like Circumboreal Forest because, as usual, the name says what we need to know, right? So circumboreal, circum means around, and borealis means the north. So around the north. This is the forest that is around the north. And well, I just showed you where the North Pole is in this map, right in the center. And so the circumboreal forest occurs around the north, okay? And the circumboreal forest is the largest forest region in the entire world. So the Russian side of the circumboreal forest is about 12 million kilometers squared. And the Russian side alone is larger in size than the Amazon forest. Just the Russian side of the circumboreal forest alone is larger than the Amazon forest. And then there's the rest of it is on the American continents. And so you have Canada that has about 30% of the world's boreal forest, circumboreal forests. So, okay, to get you oriented on this map, this is looking, so you know how the world is a circle and there's the North Pole on the top and the South Pole on the bottom. In this map, we're looking down on the North Pole. So the North Pole is here. The North Pole is here. So we're looking straight down on the North Pole, which means that we would see the things that occur in the north, right? Like Russia and Canada. Here's Alaska, right? Here's Greenland. New York, New Jersey is not even on this map. It would be down here somewhere. So that's what we're looking at here. Yeah, looking down on the earth. So we're used to seeing the earth from the side, as it were. We're looking down on it at the north part of the earth. Yeah, so circumboreal forest, super, super big. And this has a tons of implications when it comes to getting carbon out of the air. So because these forests are so big and are larger than the Amazon rainforest, these forests contain 5 times as much carbon as the temperate forest and 2 times as much carbon as the tropical forest. So what does that mean? Remember that plants take carbon dioxide out of the air and use it to make sugars. So in order for all these plants to grow, they would have had to take carbon dioxide out of the air and turn it into sugars. So because of the sheer size of these circumboreal forests, they contain a lot of carbon compared to other forests in the world. And you see here, it says associated peatlands, right? Associated peatlands. Remember way back when we were learning about the mosses, we learned about peat moss. Near these forests are also a bunch of those peat bogs, which we learned about way back in the moss lectures. All right. Conifers are important. They're important, as I just mentioned, for removing CO2 gas from the atmosphere. So that's super important. But they also have a lot of economical importance to humans. So we use conifers for lots of things. We use conifers to build things, so lumber, right? Pine floors, houses out of different types of conifers, etc. The paper industry actually uses a lot of conifers. In fact, unless you buy recycled paper and unless you buy recycled toilet paper, and even sometimes then also, but a lot of toilet paper is made out of conifers, believe it or not. Furniture, yes. Fuel, yes. And then other products. So if you've ever used turpentine, for example, that comes from conifers. If you ever saw Jurassic Park and they like had the mosquito in that weird rock, and then they got the DNA from the mosquito in the weird rock. Well, that weird rock is amber or fossilized resin from conifers. And then some food products. So for example, pine nuts. So if you've ever eaten pine nuts, those are from conifer cones. And you should note that conifers can't make nuts. Only angiosperms can make nuts. So when it says pine nuts, that's not botanically accurate, right? Same thing goes for ginkgo. I think sometimes they say ginkgo nuts. No, they're not nuts. Stop trying to use words from other plants. All right, new word today, xerophyte. This is a new word. So conifers are xerophytes. What does that mean? Well, xerophytes or xerophytic plants live in dry areas, live in dry conditions. So the word xeros means dry. And we already know that phyte means plant. We already know that. So xerophytes is a plant that can live in dry conditions. And think about it. So I just told you that conifers live in those circumboreal forests, which are in the north. And it's cold. It's cold up there. It snows a lot, right? The temperatures tend to be very cold. And so, yeah, like there is a lot of snow around up there in the north. But when the water is frozen, it's effectively unavailable to plants when the water is frozen. When water is frozen in the form of snow or whatever, plant roots can't suck it up, right? So even though they're surrounded by snow, they're basically, it's very, very dry for the plants because they can't access the water because it's frozen. In the New Jersey area and other places on Earth, conifers also live in very sandy environments. So that's what's showing you here on the right, in the very sandy environment, which also happens to be dry. Alrighty. Okay, so I just told you the conifers can live in these really dry environments, right? Either in the north where water is frozen or in sandy environments, maybe, for example, next to the seashore. So how do conifers live in these environments? The way that they do, the main way that they do is because of their leaves, Their leaves over evolutionary time have become adapted to deal with these xeric or dry conditions. They've done this in a few different ways. They've done this in their shape and also their anatomy of cell structure. So first we'll talk about their shape. So many conifers have this type of leaf, right? A needle-like leaf. So like if you think of like a Christmas tree, for example, it has those kind of needle-like leaves. Other types of conifers have these more scale-like leaves. So it almost looks like the skin of like a snake or something. So just to get you, just so you know what I'm talking about here. I guess I'll use bright blue. Each of these little things, like here's one, is a leaf, right? So very small and needle-like. Here, each of these scales is a leaf. So you can barely see them because they're so small, but okay, like here's one, right? Here's another one. Very small, scale-like leaves. Other conifers have what's called an awl-shaped leaf or awl-like leaf. So awl might be a new word for some of you. It's not something that we say that much anymore. Awl is a tool that is used for leather working. So if you've ever made shoes, that's one application, or worked with leather to turn the leather into like a jacket or something like that, you would have maybe used this tool called an awl. And so that's what we mean when we say awl like leaves. It just shows you how that's what somebody would have named this shape, probably when a tool like this was a lot more relevant in day-to-day life, so that everyone was like, oh yeah, an awl. But now it's like kind of like, what's an awl? What? I don't know what that is. Oh, there you go, got a picture. That's an awl. So needle-like leaves, scale-like leaves, awl-like leaves, and some more laminar leaves. So remember, the green part of a leaf that we're used to, right? Like if you see a simple leaf, here's a leaf, right? Simple leaf. This part is the petiole. This part is the blade or the lamina. So that's where this is like saying laminar, meaning kind of flat and green, flat and green. So it's hard to tell in this photo because this kind of still looks a little needle-like. But trust me that these leaves that's shown in this photo are a little bit more flat and broad than the regular needle, which is like very pokey and thin. So why am I telling you about all these leaves? Because remember, these are ways that conifers are able to live in these dry environments because of these leaf shapes. It's one reason. I think I might say, yeah, so these these 4 leaf shapes, right? Needle-like leaves, scale-like leaves, awl-like leaves, and even laminar leaves. The main takeaway is that the leaves have reduced, I'll write it in the chat box, reduced surface, if I can type, area. The leaves have reduced surface area, so there's less leaf to be exposed to the air. And so then there's less water being lost from each leaf because the leaves have this reduced surface area, and that helps them live in these dry environments. In addition to this reduced surface area, conifer leaves also have their stomata sunken into the leaf. So on this drawing, oh, I probably show you, but here, let's just highlight one down here. So look over here. See this kind of pit? Here's another one here. Here's another one here. Oh, and there's 2 more over here, right? 1 and 2. So in these little pits, the stomata are actually going to be located on the bottom of those pits. And what that does is it also helps with water loss. So think about it this way. Let's say you're in the north and it's cold and you're covered in snow and it's really windy, like today here in Michigan, super windy. And wind's blowing on you and it's so cold. Well, when wind blows on plants, it's gonna remove water from the leaves more readily. So for example, like if you have a house plant and you have a fan and you put the fan on the house plant all day, it's going to need water more than if you didn't put the fan on the house plant all day, right? Because it's just going to cause water in the soil, but also water in the plant to just be evaporated. Well, if your stomata are sunken down into the leaf, then when wind goes by you, like, so let's say this pink is wind. Let's say this is wind, you know, blowing. I love it. Here's wind blowing, you know, along the surface of the leaf. Well, if your stomata are sunken down inside there, it's almost like you put curtains or something on your window, right? Like in the winter time, it gets really cold, and like maybe if the wind's blowing, kind of gets drafty in your house, maybe put a little like curtain on your window, and then it kind of like helps it a little bit. Anyways, so conifers can live in these dry environments like the north where it's frozen, or sandy environments. How do they do that? Number one, they have reduced leaf area in those shapes we saw. Number two, sunken stomata. Here they are. And also good on me for finding all of these so quickly. Here you go. So in case you were tuning out just now and not listening to me, which I know none of you ever do, here's the reasons why that conifers can live in those dry environments, right? Reduced surface area. In addition, they have this thicker cuticle than the other plants we've seen so far. Sunken stomata, a hypodermis. Don't worry about that too much. Oh, no spongy mesophyll. Okay, right, no spongy mesophyll. So we haven't really gone into the anatomy of some of these. Here, let me get out of this. You know, we've talked about mesophyll as a ground tissue in the leaves, but we haven't really talked about the different types of mesophyll. Suffice it to say, normally there's two types. Usually there's like a palisade mesophyll. Oh yeah, we did talk about it, I think. Can't remember. And spongy mesophyll. Those are two types of mesophyll. Mesophyll is a tissue that's inside of leaves. And in the case of conifer leaves, there's no spongy mesophyll. They only have palisade mesophyll. That's part of the reason why they're able to have a reduced size, because they've gotten rid of some of these tissues, including the mesophyll. Oh, on the next slide, I had mentioned the endodermis, which is this layer here that helps prevent water loss, but not super concerned about that. Although check out this thicker cuticle. So like it said, thicker cuticle, much thicker cuticle than we've used to see. Right, so yeah, these are the ways that they prevent water loss. Hypodermis and endodermis, don't worry too much about those, but just that they reduce. Oh, here you go. Yay. That's good. Oh, sorry. Yeah, endodermis is the ring in the center that I drew. Sorry, hypodermis, it's closer toward the outside of the leaf. But anyways, here you go. Here you go. This is a cross section through a conifer leaf. This could come in handy. This slide can come in handy when you guys do the conifer lab. Okay. How are we doing on time? Oh, it's just ticking away. Resin. What is resin? Resin is this sticky fluid. So, okay, have you ever smelled a conifer? Like, if you've walked around during the holiday season and there's Christmas trees around, and maybe you walk by somebody selling Christmas trees and you like smell the smell of Christmas trees, or maybe you've bought for your car like an air freshener and it's like in the shape of a little Christmas tree and it has that pine smell, or even maybe you've used pine sol, right? Different things that have this like kind of evergreen pine smell to it. Well, the reason why it smells like that is because of this liquid called resin. So resin ducts are like tubes. Here's one. Here's one. Sorry, my stomach is like making noises today for whatever. I guess I didn't eat enough breakfast. Resin ducts are a tube, and in the tube is resin. Resin is a sticky fluid that is inside of the pine or the conifer trees and in their leaves as well. And what that does is it not only does it help it, so I think it was on the previous slide as well. Let's look. No. Okay. What it does is basically it helps prevent fungal infection of the tree and leaf, particularly the leaves. It also makes insects and other animals not want to eat it because it doesn't taste very good. And so it helps prevent insects from infesting the trees and other animals from eating the trees. Although deer, for example, when it gets to like the thick of winter and it's super cold out and there's not any real leaves around, like they will end up eating evergreen conifer leaves, but it's not their preferred food choice. Resin is also how we get amber. Resin is amber. So maybe remember at the top I mentioned Jurassic Park and like, oh, look, there's a mosquito in this weird rock and we're gonna like extract the DNA out of this mosquito and we're gonna use it to like make dinosaurs, you know, that kind of thing. Yes, so this weird rock thing is amber, and what amber is it's fossilized resin. Fossilized resin. So here we're talking about resin, here we're talking about fossilized resin. And sometimes, if you're lucky, sometimes insects and other small mammals or small animals can get caught in this resin way back in the, you know, millions and millions and millions of years ago. And then the resin fossilizes, and it's cool because you have a really good specimen of animal trapped in that resin. People also use amber to make jewelry. So if you've ever bought an amber jewelry, you're buying fossilized resin jewelry. Okay. Most conifers have a taproot system. Taproot system, I think, came up in a previous class, but just, you know, taproot means like the main root is kind of long and deep, right? So for example, a carrot, it's a type of taproot system. I think maybe yeah, here we go. So most conifers have a taproot system that looks like this. So long root, and then with with some lesser roots kind of coming off of it. But one main long root, that's a taproot. Oh geez. Taproot. Other plants have this kind of fibrous root system where there is not any main root. Instead, there's just like lots of roots coming off of the base of the shoot system, but no main one. That's your fibrous root system. But most conifers have taproot system. Oh, symbiotic relationship alert. Wah, wah. So most conifers also have this symbiotic relationship with the mycorrhizal fungi, which helps them gain nutrients and minerals from the water, from the ground, and the fungi in turn gets glucose from the conifers. You might remember way back when we watched that first documentary, What Plants Talk About. There was a whole section about mycorrhizal fungi in Douglas fir. You might remember that, maybe that's reminding you. And that's what we're talking about here, is that fungal association, symbiotic relationship with the conifers, and the fungi provide the trees water and minerals, and the tree provides the fungi glucose. That's what we're talking about. Okay, gonna have to talk a little bit about growth, just a little bit. So this is similar to what we saw already, in the cycads and in the ginkgo. So some of this is going to be a review. If you're looking at part of the plant that is younger and that still has primary growth, you're going to see your typical players of epidermis, cortex, the vascular tissue, which is going to be arranged in the eustyl, and the pith. But since we're in the gymnosperms now, in particular the conifers have abundant secondary growth. So ginkgo has abundant secondary growth, conifers have abundant secondary growth, cycads have limited secondary growth. And we already have learned these other things here: 2 lateral meristems, vascular cambium, cork cambium, Vascular cambium is producing secondary xylem and secondary phloem. Cork cambium is producing phyloderm and cork, which we've already covered. So here is a cross section through a conifer stem that is on its way to producing only secondary growth. So if I highlight some of these, let's use red. So we already see secondary xylem, right? Look, here's one ring. Here's another ring, the secondary xylem. So, whoa, right? Can I draw that? Yeah. So we see secondary xylem, so that already exists. And we see secondary phloem up at the top, right? Which is around here. We see the vascular cambium, which is the intersection. So I'll use a different color. Let me use a different color for that. I'll use yellow. So this is that lateral, one of the lateral meristems, vascular cambium, producing secondary xylem toward the inside, producing secondary phloem toward the outside. But this still has pith, like we still see pith. We still see pith here. So we're still transitioning from primary tissues to secondary tissues. And I'll tell you, pretty much anytime you look at a microscope slide of a conifer, you're usually still going to see some primary structures. Why? Because whatever specimen you cut to fit on a microscope slide has to be relatively small, right? Like when I showed you at the very beginning of the class, that person sitting next to that big trunk. Well, as much as I'd want it to, that big trunk cannot fit on a microscope slide, right? So normally if you're putting something on a microscope slide, it's pretty thin and small. And so yes, you do have these secondary tissues, but it's still gonna have some of these primary tissues just because of the size, whatever, yeah. It has to fit on a microscope slide, so it's gotta be, yeah. Anyway, so just so you see this cross section here. Bark, I don't know if we've come across bark before, but if we haven't, don't worry. It's another collective term. So we had the term paraderm. I'll write it in the chat box. We had the term paraderm, which was the cork or phellum, the phyloderm, and cork cambium. So we have this term paraderm, which was a collective term for these three tissues. Bark, which you've probably heard the word bark before, right? Look at this tree bark, right? Not bark like, ruff, ruff, not that kind of bark. Tree bark. Actually, for your phenology assignment, you're taking one of the first observation day, you were taking photos of the bark in addition to the rest of the tree, right? So bark is another collective term, and bark is basically the paraderm plus the secondary phloem. So look here at the bottom, it says bark is a collective term that refers to everything outside of the vascular cambium. So I already showed you where the vascular cambium is, right? Here it is, and it continues all the way around. I'm not going to draw the whole circle because you know how great I am at drawing. And so bark is going to be everything here. What does that include? Bark equals the paraderm plus secondary phloem. Or another way you can say is bark is equal to the cork, the phyloderm, the cork cambium, and secondary xylem. Yeah, that's what bark technically means. So if somebody's talking to you about bark, that's what bark is. Okay. Botanists love collective terms. I don't know why. I guess it's, I don't know why. I don't know why. All right, so we're talking about conifers. In the conifers, the primary phloem and, the primary xylem and secondary xylem in conifers are composed of a cell called a tracheid. And we call conifers softwoods because, here's tracheid, because they don't have fiber in with their xylem tissue, whereas when we get to the angiosperms do have fiber, and we call them hardwoods. But this is why we call conifers softwoods. So the primary and secondary xylem are composed of tracheid cells. The phloem, primary and secondary phloem, is composed of sieve cells. So tracheid cells are xylem, sieve cells are phloem. Just like the xylem does not have fiber cells, the sieve tube cells don't have sieve tube elements, which we will talk about later. Talk about that in the angiosperm lecture. Okay, so I'm going to walk you through these slides. These slides will be helpful for you when you do the conifer lab, because depending on how you cut through the trunk, you're going to see different cells. So if I were to use like an example in like a human body, right? Like here's my arm. If I cut my arm like here and you were able to look at it, you would see in the middle like a bone and then stuff around it, right? But like if I put it this way, it's going to look different. It's going to look different, right? Or like, what's it better? Here's this water bottle, right? Here, no advertising. Here's this water bottle, right? If I cut the water bottle lengthwise, well, I guess there's really not much in a water bottle. That's not a good example. But anyways, the point is like, depending on how you cut the thing, you're going to see different things inside. And that's what I'm trying to get across here on these slides. So this is a piece of trunk or stem from a conifer. And what we're mostly seeing here is the secondary xylem. So the stuff that there's a ring every single year, and makes up the bulk of the stem once you're in secondary tissues. And if you just cut the tree down, right? Like you just cut the tree down, maybe you have a stump somewhere near your house or apartment and you could like, you've seen a tree stump before. That's like cutting the tree down. And if you were to look at that tree stump, here's what you might see. This is what the cell structure would look like. However, if you took the tree that you cut down and then turned it into like, cut it in half, like long ways. So like if you, here's cutting the tree down, like here's the tree, cut it down. And then this is cross section, right? Cross section of a tree. But if I were to cut it this way, right? It may look more like this, tangential section. And this will be, I'm going to show you on the next slides, and it'll make it more clear. But let me just show you the different sections for right now. So cross section. Oh, and for you guys going to medical school, you have to know these things. You have to know these things. So it's not just me. Like I didn't make these up. So cross section, tangential section, radial section. The three different views, depending on how you cut through the stem. All right, so this, let's see. I'm going to go back for a second. I'm going to go back. So here, remember, let's focus. So we're going to focus on the secondary xylem, and we're going to focus on tracheids. Look, tracheids, right? Secondary xylem, primary xylem, but let's just focus on secondary xylem and let's focus on tracheids. So here we go. This is a piece of wood which is composed of a tissue called secondary xylem. And that means that there's going to be a bunch of tracheids here. If we were to do a cross section, so here's one tracheid. Here's another one. If you were doing a cross section, you would just see the tops of them. So like, here you go. These are the tops. That's cross section. All right, come on. See, cross section. If you're going to do a tangential section, you would see them long ways. Here's one. Here's another. And if you were going to do a radial section, you would see the sides of them, but you would also see the wood rays. So like, here's a wood ray going that way. Okay. Here is a cross section through a tree trunk. So just to get you oriented. So this cross, all right, sorry. This is a cross section. So when you see a tree like this, you're seeing this. These are the cells that you're seeing, right? So for example, look, rings. Look, early wood. Look, late wood. Don't worry about where they're oriented right now, but look, these are the tree rings, right? Tree rings. Early and late wood, tree rings. This is a cross section. This is a cross section. If you were going to do a tangential section, I don't know if you remember from, what is it? Trigonometry? No. Calculus? Probably. A tangent, I don't know if you remember what a tangent is, but basically if you have a circle, like here's a circle. Tangent is a line. Oops. Tangent is a line. Oh geez, I missed it again. Like here, this is a line that's tangent to the circle. This is a line that's tangent to the circle. So the point is, if you were to do a cross section, so like, look, here's a line that's tangent to the circle. Can I grab that? Maybe I can grab it. No, thought maybe I can grab it. If you were going to do a cross section through, sorry, a tangential section, let's say like this through this tree, you would see the stuff that you, oh geez, I'm getting in the weeds here, kids. You would see this, you would see those cell structures. And then lastly, radial section, just so we know what we're talking about here, where you would see the wood rays. Remember, the wood rays radiate out. See all those lines radiating out along the radius of this. So if you're to do a radial section, that would be like cutting the tree like this. And then you would see those cell structures that we saw on the previous slide. Whatever. Let's move on from this. Yeah, so this slide is just telling you about the wood rays, which I just drew. I'm going to move on from this. Bye. Moving on. All right. Conifers, let's see where we're at here. Yeah, okay. Conifers are usually monoecious, right? Sex structures, both male and female sex structures on a single plant individual. And there's some repeat players here, and then some new things that we have to learn. So the male reproductive structures are strobili with microsporangia that produce pollen grains, just like we saw in the cycads, just like we saw in ginkgo. Same thing. Cycads, ginkgo, conifers have the same male reproductive structures. Strobili with microsporangia that produce pollen grains. However, we're in the conifers now, right? Conifer is to bear cones. And so now we have these new structures called cones, and the cones are where the ovules and later the seeds are located. So here you go, I've color coded it for you. Got male reproductive structures. So there's a bunch of strobili down there on the bottom. You can see many of them there. And then I've circled one cone here in red. Okay, let's talk first about the male. So Male reproductive structures are strobili that have on them microsporangia that produce pollen grains, right? So here on the left, you see many male strobili and pollen being released. Oh, and I can confirm, so Ben did send me the link we were talking about last time about mass planting of male trees and how that could aggravate allergies. So Ben did share some links with me, and yes, it does appear that gymnosperm pollen can cause allergies in some people, although it tends to be, I think, less common than angiosperm allergies. Anyways, I told you I'd follow up on that, so I'm following up on that. Thanks again, Ben. But look, so yeah, so here you go. Pollen grains being released into the wind. Here's the close-up of the female reproductive structures. There's on the left, you have a cone that's been cut through longitudinally, and you could see the ovules. Do I color the ovules? No, here. So this is one, here's one oh, yellow's not good. I guess blue. Here's an ovule. Here's an ovule. There's another one. Later, those will become seeds. Here you go. Here's a bunch of seeds. Here's a seed. There's another one. Later, after fertilization occurs, the ovules will be seeds and they'll get released. All right. The agent of pollination. Agent of pollination is something that we've come across before, right? I told you it's not like a James Bond movie. It's the way that pollen gets from the microsporangia or pollen sac to the ovule, right? The agent of pollination. And in the case of the conifers, it's wind. Wind is also the primary method of seed dispersal. So how do seeds get from a tree to the ground? In conifers, it's wind. On the left here, you can see this is a bunch of male strobili. Each of these is a strobili. And look at how much pollen. Look at all that. That's all pollen grains being released from the wind. Being released into the wind and flying away, right? Flying away to go pollinate some ovules somewhere. And here you go. Here is a close-up of one pollen grain. So this whole structure in this right side is a pollen grain. And the pollen grain have these cool little air bladders. So here's one. Here's another one. Like cool little air bladders that help them stay in the air longer so they can travel farther distances. And the air bladders also help orient the pollen grain toward the ovule in the correct way. And one thing that Dr. Casper always pointed out is like, what does the pollen grain look like? Well, here you go. You got your air bladders, and okay, so you got two air bladders and then a circle, and then, oh geez, here you go. So you always said it looks like Mickey Mouse. Yeah, I know. So I can never forget that when I think of it. All right, so here's a pollen grain. Look, it kind of looks like, look, here's Mickey. Look, here's a circle. Here's an air bladder. Here's an air bladder. Look, this one even has little, like, you see the cells inside kind of look like, like screaming Mickey. But yeah, so you see two pollen grains here on the left. If we were to, yeah, I know. It's like every time you see a pollen grain from conifer, you're always going to think Mickey Mouse now from forever. So yeah, on the left you have two pollen grains. On the right you have one of them. This one here is under SEM microscope, so you can get an idea. And then on the top here is diagram showing you the components. Same things we've already covered, right? Same things we already covered. We have two prothallus cells, which we still don't know what they do. We have pollen tube cell, and then the generative cell that's going to produce sperm. So this is all the same stuff we've seen before, except for the air bladder. The air bladder is a new thing. Once the pollen fertilizes the ovule, the egg in the ovule, we'll have a seed. And so what's cool is the pollen grains have air bladders that help them fly. Well, the seeds also fly. The seeds also fly, and and some of them have wings. Some of them have wings. So here is a seed. There's another seed. And this flap is the wing. Flap is the wing, and that helps them fly. It helps them stay in the air longer so that they can fly further distances. So some species of conifer have weird cones. Oh, Brittany asked, are there multiple pollen grains produced in each pollen sac? Yeah. Yep. Just like, right, because inside of a pollen sac, remember, pollen sac is another word for a microsporangium in the gymnosperms. Each microsporangia produces lots of microspores inside of it, and each of those microspores will go on to become a pollen grain, which will get released. So yeah, multiple pollen grains per microsporangia, aka pollen sac. Yes. So some conifers have weird looking cones that are almost fleshy in appearance. You may have seen, I know me, when I was growing up in New York, I remember seeing these as a kid, and like grown-ups telling me, like, don't eat that. And you still shouldn't eat that. But yeah, so there's some species of conifer produce these kind of weird fleshy cones, and these, when they become seeds, are going to be eaten by birds. So the reason I'm pointing this out to you is because most of the time the seeds have wings and fly away. But other times, if the species produces a fleshy cone, birds will eat those fleshy cones and poop out the seeds later. Yeah. All right. Alternation of generations, but they're not safe for us to eat, right? Yes, please don't eat these. No, yeah, they're not berries, I know. And so they look, with the eye, they look, I know, don't eat them, guys. With the eye, they look like berries, right? In fact, we'll learn this in the next class, but in the case of these ones, these are a type of juniper cone, and people actually do call these juniper berries, which is not right. It's kind of like a whole pine nut thing. Oh yeah, I'll just put some, oh, this is a Brazilian sea bass with roasted pine nuts. It's like, okay, it's not a nut. It's from a pine tree. It's from a cone. It's not a nut, but people call it pine nuts. And people call these things I just circled here juniper berries, and we actually use those to make gin. We'll learn about that in another class. All right, I got a whole bunch of things here in the chat box. Whoa. I gotta slow down. Let's see. Yeah, don't eat them, please. Yeah. So do they have more than one ovule? You mean these fleshy cones, Brittany? Yeah. In terms of the fleshy cones, they usually only have one. So if you go back here, you can kind of see inside of there is a single ovule. In the case of these quote unquote juniper berries, you can't really see the ovule, but in the fleshy cones, there's usually just one ovule per fleshy cone. Whereas in the woody looking cones, there's usually lots of ovules. Yeah, but birds eat these, birds poop them out, and then the seed will grow somewhere else. I think we're almost done here. Yes, we are almost done. All right, good. We're going to finish. That puts us in good shape. So alternation of generations in the conifers. It's the same process, basically, that we've seen in the other gymnosperms, right? So the cycads and ginkgo. Same process. For the most part. One difference now is that the pollen grains have air bladders, right? Another difference now is that the female sex structures are arranged in cones. Those are two differences, but in general, same structure, same process. So here, this is just your convenient little overview slide of alternation of generations and conifers. Obviously, there's some words on this slide that you don't need to know. So if I haven't gone over it in lecture, you don't need to know it. But if we have gone over it, you do need to know it, right? I will show you a few things here, but let's just say, note that, I guess let's use red. Right, here you go, adult sporophyte, 2N, adult sporophyte. So this whole giant tree is your sporophyte plant, right? Your sporophyte plant. And on that tree, you're gonna have microsporangia and you're gonna have cones on that tree. So let's go through it just a little bit, right? So here we go. We just zoomed in on the microsporangia that are arranged in strobili, right? Here, I'll use, well, I guess blue, right? Here's one Strobilus, right? Here's another Strobilus. Here's a whole bunch of Strobili. On each of those, on each Strobilus, right? You have microsporophylls that have on them microsporangia. Inside of each microsporangia, there's going to be microspore mother cells that'll ultimately undergo meiosis, right, to make the microspores. Down here on where the female reproductive structures are located, you have cones. So here's part of a cone that's already finished. Here is a little immature cone that's on its way to becoming a cone. And on those cones are these scales. So on cones, there's all these different scales, and we'll look at what that is on another, but those are, if you're looking at a woody brown cone, all those little like pokies that are coming out, those are the scales. And we say it's an ovuliferous scale because it's that same root word. Fer means to bear. Ovule refers to ovules. So it's a scale that bears ovules. But scale is fine. You just know the word scale. But you might see this ovuliferous, but scale, just know scale of a cone. Scale of a cone, each scale is going to have on it usually 2 ovules. Each scale. Yeah. Okay. Here is a close-up of a male Strobilus. This is what I was saying before. We're gonna, like, a Strobilus and a cone are 2 different things. Strobilus and a cone are two different things. Male Strobilus has on it a stem, right? Leaves, here's a leaf. And each leaf is going to have on it, or I should do this. Stem, leaf, here's another leaf. Here's another leaf. And on the bottom of each leaf is a microsporangia, right? That's what Strobilus is. Yep. Whereas cone is not the same. Cone is different. Cone has, do I say it on the next? Yeah, good. All right, so this is here on the left, you have a cone, right? This is one cone. The next one in the middle, that's a longitudinal section through that cone. And it shows you all of the different scales. Here's a scale. Well, not all of this is a scale, but the point is, look at all these pokey things coming out, right? Part of that pokey is a scale. So we're going to do a close-up, and that's what we see on the right here. That's what we're going to see on the next slide as well. So cone is a modified branch system. It's composed of a stem in the middle. Oh, I should use a different color. Hold on. Sorry, I accidentally muted. I guess I'll use yellow. So you have a stem. Here's the stem. Then you have leaves. Here's a leaf. Here's a leaf. Then you have scales. Here's a scale. Here's a scale. And each scale is going to have usually 2 ovules on it. In this slide, you can only see one. There will be another one directly next to it that you can't see on the top surface. So this is why a Strobilius and a cone are different, because a Strobilius is a stem with leaves on it. A cone is a stem with kind of more stems on it and leaves, because the scales are basically like modified branches. Don't worry about it too much though, but this is why I make a big deal about how cycads don't have cones. Only conifers have cones. This is so you have it for when you're referring it to it later on your own. Here's a nice cross section. This will help you in the lab, right? You have the scales. Here's an ovule right there. Oops. Clear. Cool. And this is just to continue on of the alternation of generations. On the top, you have a pollen sac or a microsporangium. On the bottom, you see an ovule. With both of these have spore mother cells, right? Microspore mother cell, megaspore mother cell, which is 2N. That's going to undergo meiosis to produce your tetrad of microspores in the case of the microsporangia or pollen sac. And then the process of ovule development. Remember, we had initially the 4 megaspores, but ultimately you only have 1 functional megaspore, right? Which you can see here. So look, so we had meiosis occur. So if we have meiosis occur, we go from a diploid thing to a haploid thing. In the case of the microsporangia, which contains microspore mother cells, after meiosis occurs, you have a tetrad of microspores. Here's a tetrad. Each of those microspores is going to undergo the processes that we looked at in a previous lecture to grow into pollen grain with its parts. Process of meiosis is going to result in 4 megaspores, but remember, this is the ovule. But remember, we only end up with 1 functional megaspore, which we've covered in previous lectures. But ultimately, you end up with the ovule, which contains inside of it many things, including the archegonia with the egg. Eventually, pollen grain is going to land on the outside of the ovule. Pollen tube cell is going to grow down. With it, the sperm are going to be able to enter the archegonia, right? This is female gametophyte plant, the archegonian egg. Ultimately, egg and sperm are going to come together, process of fertilization, and the result is a 2N zygote. So these slides might look complicated. It's just because they're putting so much information together, and we've already covered this information in the previous lectures. In the cycad lecture, in the cycad lecture and in the ovule lecture, we covered all of these processes. So if this slide is looking super complicated to you and you have no idea what I'm talking about, go back to that ovule lecture and go back to that cycad lecture where I detail these things out to you. The only difference now is we have a scale, right? That's really the only difference, is that we have a scale now, and the pollen grains have air bladders. Don't worry about, there's a lot of stuff on here you don't need to worry about. The point is, you had egg and sperm come together for 2N zygote. 2N zygote's going to grow via mitosis, and ultimately this is what you end up with, right? Oh, yeah. Ultimately, you end up with a seed with embryo inside. And then there's a few words here, like these are parts of the embryo. Cotyledons are the embryonic leaves. shoot apex of the embryo is going to be where the apical meristem is on the embryo. Hypocoidal we covered before, it's a part of the stem. Root cap and root apex, those are all parts of the embryo. Embryo is surrounded by female gametophyte plant, which it will eat to grow. Embryo is protected by seed coat. We've covered that before. And then eventually the seed is going to get released. So here you go. This is a seed getting released. Here's its little wing. Here's its little wing. All right, I think we're basically done here. Good. Yep. So here you go. Seed gets released, lands on the ground, germinates somewhere, and then you have the embryo can keep growing and grow into a mature sporophyte plant. There you go. Yep. Cool. All right. I think that's it. That is it. Wow, we did it. Man. Stop sharing this. Cool. So yeah, so we covered that's conifers. Next time, so some of these things that we talked about today will also come up next time. Let me pull up the syllabus real quick. What's today? Today's the 30th. So today we covered the conifers. And you had an assignment due before class began. So again, if you didn't turn in your assignment number 5 today, please email me so I could reopen it for you. Next time we're gonna have like a virtual field trip, as it were. So what I'm gonna do is I have pictures of trees that are located on campus, New Jersey Institute of Technology and Rutgers Newark. And we're going to look at those different trees and talk about the structures. And so there'll be some repeat information from what we saw today as we go through those different species. The following class, we're still figuring out what we're going to do with that. But basically after the virtual tour, then we're going to go right into the angiosperms and finish up the rest of the semester. So, and a reminder that your next lecture exam is 2 weeks from today. So stick with it. We're getting there. We're almost to the end. We're almost to the end. So thanks so much for joining me today, guys. And I will see you on Thursday. Sure.
Stay safe, Professor.
Bye. Thank you. So somebody has a question?
Yeah, I do.
Okay.
The assignment that we just did, is that grade accurate? Because I think we had to type in something. So I'm not sure if you're like going to grade it and then change your grades for it, or how does that work?
So as I've said many times in this lecture, like since the beginning of the semester, when you fill out an assignment on Blackboard attempts to grade it. But I go in and I manually grade every single assignment. So, yeah, the grade that you see right now is not probably accurate because I haven't had a chance to go in and grade it yet. So every assignment that is submitted through Blackboard, I will manually grade that. And so, yes, stay tuned for the actual grade.
Thank you.
Yeah. Any other questions?
Hi, Professor.
Hey, what's up?
I can understand how the ginkgo seed, it looks like a fruit, but it's a seed. But how is that berry a cone? Or how is that thing that looks like a berry a cone?
Right. So fair question. It's a cone. The simplest answer is it's a cone because it's on a conifer. It's a cone because it's on a conifer. To be fair, it doesn't look like a cone at all. But what would have happened over evolutionary time? Let's see if I can do this here. Let me pull this up. Yeah, here, I'll share this. Where are you? I lost you guys. Here we go. It's a fair question. Yeah, I'll make this big. So what would have happened, Brittany, is that over evolutionary time, most likely due to pressure from things eating it, but what probably happened is this. So these scales, all of these scales and their associated leaves and things, okay, actually, here's a better way to describe it. Remember how I said this is a stem, right? This part in the middle here is a stem.
Yes.
Right? So that means that way back when over evolutionary time, it may have looked like a more normal stem, like a stem of a tree. It may have looked more like a normal stem that would have had like a scale here and a scale here and maybe another scale way down there. But over evolutionary time, you can't fit, if you're on one regular branch, you can't really fit that many scales on a branch. So what would have happened is the branch would have gotten shorter and shorter and shorter and shorter so that it could accommodate having lots and lots of scales, such that you end up with a pine cone, right? For those species with those fleshy cones, additional processes would have occurred such that each of these things, let me clear all the drawings, each of these things, so here's one scale with a leaf and an ovule. Remember when we learned about how the ovule came about, like back in the introduction to the ovule class, where it was like, oh, you probably had like an ovule just like out, and then eventually like the tissue around the ovule would have like surrounded it to make the integument. That's kind of the same thing that would have happened here with those fleshy cones. So like instead of there being a woody scale, for those species, the leaf tissue and such got modified such that it kind of just surrounded the ovule and looks like a fleshy little thing. But
okay,
yeah, don't get too tripped up about it. It's the point is that most conifers have like what we would just call a pine cone, right? Like a pine cone looking thing. Most have that, but there's a few oddballs that have these fleshier cones that are eaten by birds.
Okay, and then I guess whenever they get fertilized or pollinated, do they fall off too?
Yep. Yeah, well, you're saying so inside of those fleshy cones, right? Inside those fleshy cones, they are cones, but they look like kind of a berry, is the ovule. When the ovule gets fertilized, the structure turns into a seed, and most of the time birds would eat that. But if the birds don't eat it, it'll just fall off of the tree and just grow right there.
Okay, cool. Thank you so much.
No
Interesting
problem.
stuff.
Any other questions? If anyone has any other questions, you can email me, and I'll get back to you sometime soon. Okay, have a great day, everyone.